We are studying the etiology of a neurodegenerative disease in the Western Pacific with clinical features of amyotrophic lateral sclerosis, Parkinson's disease, and an Alzheimer-like dementia, or ALS PDC. Individuals in this region have historically been exposed to plant genotoxins through traditional food and medicine. We think that these genotoxins trigger the progressive degeneration and loss of neurons in ALS PDC brain by activating cellular processes that lead to genotoxic stress. In non-nervous tissue, genotoxic stress has been shown to cause downstream effects that trigger apoptosis, DNA repair, and the inappropriate activation of the cell cycle, as seen in Figure 1. Normally, neurons withdraw from the cell cycle, remaining in the G0 phase, and are terminally differentiated. It is likely that reactivation of the neuronal cell cycle contributes to the pathogenic mechanism of ALS PDC, as has been reported in related disorders like Alzheimer's disease and ALS. Data from these preliminary studies is expected to provide important insight into the underlying mechanism of the neuropathology in ALS PDC, as well as reveal the potential environmental triggers. For these studies, we immunoprobe brain tissue of a healthy control and patients with Alzheimer's disease with Lewy body, or ADLB, and Guam PDC. The primary antibodies we used were H2AX, a DNA damage marker, P53, a marker of DNA damage response, and cyclin D1 or PRB, two cell cycle proteins. Immunostained brain tissue sections were visualized under a microscope and analyzed for the intensity of nuclear staining. We found that all the targets were increased in the diseased brain tissues. Brain tissue that had been immunoprobed with H2AX, as seen in Figure 3, indicates that DNA damaged via double-stranded breaks were more elevated in the ADLB than the Guam brain. In addition, Figure 4 shows a more prominent stress response to the DNA damage in ADLB than the Guam brain. An activation of cell cycle proteins, cyclin D1 and PRB, was also noted in Figures 5 and 6 respectively, with more prominent activation in ADLB. These preliminary findings support a role for environmental genotoxins in the etiology of ALS PDC. Studies are now underway with additional PDC patients using other markers of DNA damage, stress response, and cell cycle proteins to confirm these findings.